Hello, morning to everyone. Welcome to Student Circle GS Foundation classes. Cell are basic, fundamental, structural unit of all living things. Cells are of different sizes. It may vary from 0.03 micron to a large cell like the ostrich egg cell, where it is approximately 170. On the basis of formation of cell, we can divide cells in two parts, prokaryotic and eukaryotic. Okay. Structurally, animal cells are little bit different from plant cells. Various cells have differences in their structure according to the need they have to perform, but the basic structure remains same. In a generalized cell, we have three major sections. The first one is outer wall or cell enclosure. It is cell wall and cell membrane. Cell wall is made up of cellulose. It is hard and permeable. Cell wall is absent in animal cells. It is present in plant cells only. Within cell wall, we have cell membrane. It is made up of lipoproteins, double bilayered lipoproteins. It is semi-permeable, means it allows selective things to enter or exit the cell. The cell wall provides enclosure to cytoplasm that contains all the organelles. And the second one is cytoplasm. Cytoplasm is thick jelly-like fluid that contains proteins, proteins, nutrients and various organelles inside, enclosed by the cell membrane. And third one is nucleus. Nucleus is the core part of any cell. It is cell wall. Cell walls are non-living. It is non-living, mostly made up of cellulose. Mostly made up of cellulose. Cell wall is porous, when it has got so many pores to allow the things go inside and outside. You can say it is permeable, means allows everything, means tiny things to go inside and outside. But this cell wall is absent in animal cells. We don't have this cell wall. It is only in plant cell. Inside the cell wall, we have cell membrane. Here it is cell membrane. Now come to this diagram. We have cell membrane. This makes boundary of a cell, gives shape and encloses all the things like cytosol and the organelles inside it. Cell wall is living. Okay, the difference, cell membrane is living. Difference between cell wall and cell membrane: cell wall are hard, non-living. Cell membrane living, soft, provides enclosure and shape to cell. It is made up of lipoproteins, bilayered. There are two layers of proteins, bilayered lipoproteins. Okay, and this cell membrane is semi-permeable. It is not fully permeable. Semi Semi means it is very specific, very selective. जिस चीज़ को अंदर जाने देना चाहेगा उसको जाने देगा, जिसको नहीं अंदर देने देना चाहेगा उसको नहीं जाने देगा. So it is selective, semi-pervasible. Only permitted goods can enter the cell and go outside the cell. Okay. The next one is inside the cell wall, enclosed by the cell wall, we have jelly-like fluid called cytoplasm. Cytoplasm is made up of cytoplasm and inclusions. Cytoplasm is very thick substance that contains proteins, lipids, nutrients, various starch and various small organs called organelle, everything inside the cell membrane. The living part of this is called cytosol, and the non-living part, these grains are called inclusions, like grains and vacuoles. These are grains, granules, grains, and this one you can see is vacuole. These are non-living parts that makes cytosol. It is also called molecular soup. The term is molecular soup. Examiner can ask you what is molecular soup. This cytoplasm is molecular soup because it contains a soup-like thing, and every enzymatic activity, every metabolic activity of cell is being performed inside the cytosol. In cytosol, we have some important small organs called organelle. This one is mitochondria. Mitochondria, the plural is mitochondrion, because a cell may have more than one mitochondria, even hundreds. If the cell requires more energy, mitochondria is power of cell, and if the cell requires more energy, it will get more mitochondria. Mitochondria, it is also called powerhouse of cell because it generates, it is bilayered, double walled, structure. You can see the wall one and inside wall. Inner wall have folds like this, angular folds. The folds, so it's called and between crystals we have open space. This one is called matrix. Open space between crystals is called matrix. Mitochondria have its own DNA. This is very specific. Normally, DNA lies inside the nucleus, but mitochondria have its own DNA and RNA. That's why sometimes we say that Mitochondria is cell within the cell. Own DNA called mitochondrial DNA. Mitochondrial DNA and also RNA, mitochondrial RNA. It generates energy in the form of ATP adenosine triphosphate. This is the universal form of energy for cellular use. ATP from food or nutrients. So it is called powerhouse of the cell. The next one we have, this is endoplasmic reticulum. It is ER or endoplasmic reticulum. It is flattened sac, tubular, as we can see. It is flattened sac like a structure connected to nucleus and to the cell membrane also. So it provides shape and support to the cell. Plasmic reticulums are of two types. ER with ribosomes attached with them and makes it bumpy are called rough ER or rough endoplasmic reticulum, and ER without ribosomes, these are called smooth or ER or SER. This one is RER, rough endoplasmic reticulum, and a smooth endoplasmic reticulum. This is responsible for synthesis of proteins, lipids, lipids from a smooth and proteins from rough endoplasmic reticulum, and lipids are from endoplasmic reticulum. The next one is, this is tubular structure arranged like a ball, and some sacs are there. These are vacuoles. Vacuoles are small sacs containing fluids that has to be carried the cell and outside the cell. So this one is Golgi body or Golgi apparatus. Golgi apparatus. The function of Golgi apparatus is to modify pack and transport proteins, hormones, enzymes means modification, packaging, transportation of protein, then synthesis of hormones and enzymes. The next one is lysosome. It's also sac containing enzymes. Better we say degenerative enzymes. Degenerative enzymes. Degenerative means it degenerates the dead parts and the foreign material that enters, foreign unwanted material that enters the cell. Because it contains degenerative enzymes to digest the waste materials and the foreign materials, sometimes it destroys its own organelles and so it becomes suicidal. So the lysosome is also called suicidal bag. Okay, things to remember, suicidal bag. Next one is peroxisome. Peroxisome. So these lysosomes and peroxisomes are absent from plant cells, not in plant cell. Peroxisome, a sac-like compartment contained oxidative substances. Oxidative, this is degenerative enzymes in lysosome. In peroxisome, we have oxidative substances. Oxidative so it is useful in lipid metabolism of lipid. It oxidizes substances, these oxidative substances, the oxidized substance, it is used in metabolism of lipids. Okay. It also maintains or reduces the level of hydrogen peroxide. The excess of hydrogen peroxide is dangerous for cell. This peroxisome maintains the level or reduces the level of hydrogen peroxide, and it also oxidizes toxins. The toxins that is coming inside or produced inside the cell are being destroyed by peroxisome. So oxidation of toxins. Okay, this peroxisome is also absent from plant cells. So, and the next one is ribosome. Ribosomes. Ribosomes are small bundles, small pockets of ribosome RNA and ribosome proteins. Okay, we can find ribosome in a group called ribosomal bundle. It can appear freely inside the cytoplasm, or it may be attached with this endoplasmic reticulum that makes it bumpy and rough Okay. So it is a small granular things, a small granular structure within the cytosol, spread over the cytosol, maybe as a bundle called ribosomal bundle, or maybe attached to this endoplasmic reticulum. Okay. This is made up of ribosomal proteins and RNA. Okay. And the its function is to synthesize proteins by joining amino acids to peptides. Synthesis of protein. Its function is synthesis of protein by joining amino acids to the peptides coming from, as per the code coming from the nucleus. Okay. We have cell membrane, we have cytoplasm, means cytosol and inclusions called. So endoplasmic reticulum, ER, rough endoplasmic reticulum, a smooth endoplasmic reticulum, vacuoles. There are so many vacuoles spread over. It carries substances basically released from this Golgi apparatus and carries them to different parts of So Golgi apparatus or Golgi body, then lysosome, then peroxisome, then ribosome. These are the granules, these are non-living parts. Vacuoles, the small sacs, empty sacs to carry water, basically the waste material outside the shell. And these are the cell pores. These sticks, small sticks, tubules are called microtubules. Microtubules. These provide shape to our shell. Macrotubules. These are macrotubules. And another important organelle inside centrioles. Centrioles. One tube, two tube, three tube, and such three structure, the 9 such structures, 3 into 9. This one makes centriole, 2 centrioles kept perpendicularly and surrounded by spindles made up of protein are called centrosome. Hence, each centriole is made up of 9 bundles of 3 tubules. Okay, 9 bundles of 3 tubules and spindles. 2 centriole, kept perpendicularly. Surrounded by spindles made up of proteins. This one is called centrosome. And this centrioles or centrosome are used during cell division. These spindles and centrioles are used during cell division. Okay. Come to the plant cell. As we discussed, the outer wall is cell wall, and the structure like this. These are plastids. Plastids. These plastids are absent from animal cells. These are present in plant cells only. Plastids are generally up to 3 types. Leucoplast, one. Second one is chromoplast, and third one is chloroplast. Leucoplast, white in color. Chromoplast have different colors, red, orange, brown. There are many chromoplasts with specific colors. Red, orange, and so many colors. There are so many chloroplasts. Chromoplast. Chloroplast are green. Chlorophyll. Yes, chlorophyll is one of the chloroplast, green. Used for absorption of heat from sun. Okay. So these plastids are absent from animal cell. The lysosome is absent from plant cell. The peroxisome is absent from plant cell. In animal cell, we have cell membrane. Living, soft, provides enclosed shape, bilayered, lipoproteins, semi-permeable. We have endoplasmic reticulum, a smooth endoplasmic reticulum, and rough endoplasmic reticulum. We have Golgi apparatus, we have vacuoles, we have lysosome. Lysosome are also produced by Golgi body. It is also lysosome is a vacuole with containing a specific enzyme, degenerative enzyme. That's why it's studied separately. It's produced from this one. Peroxisome, but peroxisomes are self-replicative. Lysosomes are produced, packed, and transported by Golgi body, but peroxisomes are self-replicative. It increases its size and then divides into 2 parts. Okay. This one is ribosome bundle, ribosome bundle. It is also in freely moving inside the cytosol, attached to the endoplasmic reticulum. We have a smooth endoplasmic reticulum, we have a rough endoplasmic reticulum, we have mitochondria, the double-walled structure, powerhouse of the cell, DNA and RNA. So it's called cell within the cell. Then we have granules, small granules, non-living things, nutrients, starch. Then macrotubules providing shape to your shell. We have centrioles, 2 centrioles kept perpendicularly surrounded by protein spindles. Centrosome used during the cell division. The difference between plant cell and animal cell, this is vacuoles. In animal cell, we have a small vacuoles, a number of vacuoles, but in a plant cell, we have single large vacuole. This is the difference between plant cell and animal cell. So we will draw a separate diagram, nucleus. This one is nucleus, because This nucleus is the master thing. It controls and coordinates. Nucleus controls and coordinates. Controls and coordinates all function of shell. Each and every function is being controlled and coordinated by nucleus through messenger RNAs. There are so many types of RNAs. We will discuss later in coming classes. So it's your structure. In next class we will pay special attention on this chromatin fiber, how it becomes chromosome and their intermediate phases.